All right, so where were we? So Gregor Mendel ended up growing 29,000 pea plants over the course of several years in a garden in his home monastery at Brno in what is today the Czech Republic, what used to be part of the Austro-Hungarian Empire. And he found that there were some traits that could skip a generation and then seemingly reappear in the next generation unaffected. Notice at the right, I've got those letters P and F. This is the standard way we refer to generations in genetics experiments. When you start the experiment, the individuals that you crossbreed make up the P generation. That P is short for parental. And so in this P generation, Mendel crossed a pea plant with purple flowers and a pea plant with white flowers. And the offspring that he got made the F1 generation. F stands for filial, which means having to do with offspring, having to do with sons and daughters in Latin. And the F1 or first filial generation was entirely purple. All of the pea plants that he got were purple. But then when he crossbred two plants from the F1 generation, he created the F2 generation, the second filial generation, and found that white reappeared in the F2 generation after seemingly skipping the F1. He never got, light purple or pink flowers. He never got spotted flowers. He never got a mix of purple and white. What he got was either purple flowers or white flowers, and the white flower trait skipped the first generation and shows up in the second, shows up in the F2. He concluded that whatever heredity was, it wasn't like mixing paint. It must be more like mixing particles. If you have a can of purple paint and a can of white paint and you pour them together, right, they blend into, light purple paint, and you can't get the purple and the white back once you've blended them. On the other hand, let's say you've got purple marbles and white marbles. You could pour both of those together in a jar, but you could still take the white marbles back out, right? The white marbles don't turn purple just because they're next to purple marbles. The color is maintained. And Mendel concluded that heredity must be like that there were some sort of little particles, some little thingies rattling around inside those pea plant cells that made the peas look the way that they do. Mendel's word for this was elements. We now call them genes. That was coined later. And what genes are is a really big black box that we have only just picked up and started to rattle. We don't know yet what these are, but Mendel argued that they do exist, some kind of little particles of heredity. Some of his particles are said to be dominant, and others are said to be recessive, because a dominant particle will cover up the presence of a recessive one. But the recessive elements can still exist. They're not destroyed, and they can be passed on, and that's the way they reappear in future generations. If you have a jar of purple marbles and you stick a white marble into the center of the jar, the white marble may be covered by the purple marbles on the outside, but you could still reach in and get the white marble back out. That's the best analogy I can think of on limited caffeine at this hour for what Mendel concluded about the way that heredity works. There's some sort of little hereditary particles. This was really interesting, but so far not that new. As far back as 1751, a guy whose name I'm not going to test you on, a French scientist named Maupertuis, had studied a family with a history of polydactyly, which is the medical term for having extra fingers and toes. Like this child's hand's X-ray shows you somebody born with an extra finger. I've never known any person that had this, but there are some lineages of cats that can actually be prone to polydactyly. There was a polydactylous stray in my neighborhood years ago. They sometimes call them mitten cats because their paws look like little mittens, and they're very good at opening things. Anyway, Maupertuis had found a family that had a history of this and plotted out their family tree and showed that polydactyly could skip generations. And Maupertuis concluded that polydactyly was caused by some sort of inherited hereditary particle, some little thingamabob rattling around inside you that makes you look the way that you do. So interesting work from Mendel so far, but at the moment it's not really revolutionary. But there was more. Mendel did something nobody had thought to do before. He counted the number of pea plants in each generation. So for example, when he crossed a purple flowered pea plant that was true breeding, meaning that all of its ancestors had been purple flowered pea plants as far as anybody cared to go back, he crossed a true breeding purple flowered pea with a true breeding white flowered pea plant. That cross is the letter P generation. All of the offspring were purple flowered. That's the F1. But when he took two of those F1 plants, we'll ignore the fact that they were siblings, and crossbred them with each other to get an F2 generation, that second generation included 705 purple flowered peas and 224 white flowered peas, which makes 929. And if you do the math, that's very close to a 3-to-1 ratio. For every 3 purple flowered pea plants, there was 1 white flowered pea plant. It's not absolutely exact, but it's close enough that it's not significantly different. If you did a statistical significance test, you'd basically find that there's no meaningful difference between Mendel's results and a 3-to-1 ratio. So that's interesting. Why 3-to-1? Why do you get almost exactly 3 purple flowered pea plants for every white flowered pea plant? And then he did some more, and he found the same thing was true for his other crosses. There were always almost exactly 3 plants with the dominant trait for every 1 plant with the recessive trait. So that's his actual data. When he crossed a pea plant with round seeds and a pea plant with wrinkled seeds, all of the F1 had round seeds. But when he crossed 2 of his F1 plants, he got 5,474 round seeds for every 1,850 wrinkled seeds. That's almost exactly 3-to-1. It's 2.96. You can see the rest of the data there. Whenever he did this type of experiment, the ratios that he got in the F2 generation were almost exactly 3-to-1. Why 3-to-1? What's so great about that? It got worse. Here's his cross of purple flowered and white flowered peas again.
Can you go back to that slide real quick?
Sure, sure. Uh, by the gods, do not try to memorize the numbers. Okay, you don't even have to memorize the crosses that he did. I'm not going to test you on, when Mendel did experiments on pea plants with side flowers, what did he crossbreed them with? End flowers. I'm never going to ask you that. I just want you to see that his actual data shows something extremely close to a 3-to-1 ratio for 7 pairs of traits that don't look like they have anything to do with each other. Like, why should tall versus dwarf plants have a 3-to-1 ratio, and yellow seeds versus round seeds also have a 3-to-1 ratio? Why does the 3-to-1 keep popping up?
You're good now. Thank you.
Okay, great. For the record, I know some of you may not be that interested in pea plants. Some of you probably don't even like to eat them. But the reason we're going on and on about pea plants is that what Mendel worked out turns out to be applicable to virtually every animal, plant, protist, and fungus, with a few added wrinkles. The same rules that Mendel worked out for determining what he would get when he crossed his pea plants determine the probability that a man and a woman will have a baby that's afflicted with cystic fibrosis or sickle cell anemia or Duchenne muscular dystrophy or any one of a number of other um, genetic conditions that humanity is prone to. Um, don't focus on the P's, focus on the general rules, because you can apply those all over the place. In fact, brief little anecdote here. When I was in grad school, um, I had this, I had a crush on a very attractive young lady who was in medical school, up a long bus ride away. So I went up for the weekend to hang out with her. And she was in med school. I got to sit in on some of her classes, and she was doing fine, and so was her boyfriend. And the important thing is that she was taking a class in medical genetics, and the students were interviewing someone with a family history of a genetic disease, something called Huntington's disease. And they were practicing taking a family history. They were practicing, trying to calculate what the chances were that her relatives were going to come down with Huntington's disease. And they were using exactly the same principles that Mendel worked out on pea plants. So this is much bigger than just peas. So Mendel looks more closely at the plants in the F2 generations and tries letting them self-fertilize. Remember that in peas, the sex organs are all wrapped up in the center of the flower. And if you just leave your pea plants alone, don't mess with them, the flowers will fertilize themselves and produce seed entirely on their own. That's not that common in the plant kingdom, but there are plants that will do that, peas being one. And so if you take the white flower pea plants from the F2 generation and you let them self-fertilize, they'll only produce more white flowered plants for as long as Mendel cared to keep on going. Never any sign that they ever had any purple flowered pea plants in their ancestry. Then he looked at the purples in the F2. 1/3 of the purple flowered plants in the F2 generation were also true breeding and produced nothing but more purples for as long as you cared to keep the experiment going. But 2/3 of the purple flowered pea plants, hey, look, computer animation, were not true breeding, because if you let them self-fertilize, they would produce purple flowered and white flowered plants in the same 3-to-1 ratio. So in that F2 generation, you're not just producing 3 purples for every white. Those 3 purples include 2 different types of purple that look identical, but one of them produces only purple offspring if you let it breed, and the other 2 produce a mix of purple and white offspring. What the heck is going on? Mendel reasoned thus. We have these little elements rattling around inside pea cells, and they determine what physical traits your peas have. Mendel just called them Elementen in German in the paper that he explained all of this in 1865. We now use the word genes. And I'm sure you've heard of genes. You hear about, genetic engineering and, you know, messing with your genes and gene therapy and gene stealer cults in Warhammer 40,000. You've heard of those. Well, the gene is still very much a black box to us. We will later get to what a gene really is, but it's going to take a couple of weeks. For now, you just need to know that a gene is just some little thingy rattling around inside a black box that determines the physical features of a pea plant. Genes can exist in several different forms, and we call those forms alleles. There is, for example, a gene for flower color in peas, and it comes in two alleles, purple or white. There is a gene for seed color in peas, and it comes in two alleles, green or yellow. There's a gene for overall size in pea plants, and it comes in two alleles, tall or dwarf, and so on. In the examples I've shown you so far, each gene has only two alleles. In the real world, you certainly can, and you very often do have genes that have maybe only one allele, not very interesting to study those, or genes that might come in three alleles, four alleles, five alleles. Last I looked, the record in humans was a gene with 26 alleles. We'll talk more about that later. For right now, you don't need to worry about it because all of Mendel's crosses happen to work with genes that only had two alleles. But in reality, there's no reason it has to be two, and it very often is more than two. Okay, why do you get that 3-to-1 ratio? Every pea plant has two copies of each of its genes because it got one copy from each parent. It got one copy from mom and one copy from dad, or one copy from the egg cell that it came from in the base of the flower, and one copy from the sperm cell that was contained in the pollen grains. So each pea plant has two copies of each genes, one from each parent, and each pea plant, if it reproduces, will pass one copy of each of its genes to each one of its offspring. Okay, with me there. The ultimate explanation for that 3-to-1 ratio is that genes come in pairs. The two copies might be the same or each might be different. Just like if you take two coins and flip them, you could either get both showing the same side, right? Both heads or both tails, or you could get a head and a tail, right? Those are your only options. When a plant's two copies of a gene are the same allele, we've got a special word for that. We call that being homozygous. Now, I'll never test you on word meanings, but zygo is the Greek root meaning to yoke together. I don't know if y'all know what a yoke is, but it was a type of harness used to attach a pair of horses or a pair of cattle to a plow. All right, if you have two oxen, you'd yoke them together so they would pull your plow or pull your wagon or whatever it is. Zygo means yoke or link, I guess, to link together. Homo means the same. So homozygous means two of the same thing linked together. When a plant's two copies of a gene are different alleles, we call that plant heterozygous. Hetero just means different, different things linked together, heterozygous. Okay, an allele might be dominant or recessive. There are some other possibilities, but we won't get sidetracked for right now. There's nothing necessarily good about a dominant allele. Dominant doesn't necessarily mean better. There are some very dire genetic defects in humans that are caused by dominant alleles, Huntington's disease being one of them. There are lots of cases where neither one is better than the other. They're just different. There's dominant does not mean superior. It doesn't mean normal. It doesn't mean common. It doesn't mean better in any way. All that it means is that in heterozygotes, where you have one dominant allele and one recessive allele, the dominant allele covers up the existence of that recessive allele. Okay, sorry, just saw something coming in on the chat. Everything seems fine. So a dominant allele will mask the existence of a recessive allele if a recessive allele is present. I should mention that dominant alleles only mask recessive alleles of the same gene. So a dominant allele for purple flower color does not cover up a recessive allele for green seed color. The two don't have anything to do with each other.
What if you get two dominant alleles?
Then you show the dominant trait. A pea plant with two of the dominant purple flower color alleles will have purple flowers. A pea plant with one dominant and one recessive will have purple flowers as well. We'll look at some cases where it's not quite that simple later, but let's not get sidetracked right now. It's not always quite that easy, but yeah, if you're homozygous dominant, then you have that particular trait. If you're heterozygous, the dominant covers the recessive and you have the same trait. Right? There's no way to tell the difference by looking between a homozygous dominant P and a heterozygous P. Okay. The pros would do this a little bit differently, but for what we need to do here, we can represent dominant and recessive alleles of the same gene by capital and lowercase forms of the same letter. So we say there is a gene for flower color. It comes in two alleles. The dominant allele is purple. The recessive allele is white. But since they're alleles of the same gene, we'll refer to them by capital and lowercase forms of the same letter. So we can call the dominant flower color allele capital P, and the recessive flower color allele we can represent as lowercase p. Now, the letter that we choose to use doesn't really matter much. You can use any letter that you want. Just like if you've done algebra problems in math, the solution is the same whether you're calling your variable x or y or q or t or d or whatever it might be. Right, the math is still the same regardless of what letter symbols you use. Same thing here. There's no particular reason we have to use P for flower color. It's just the way it's often done. In professional genetics, there'll be some standardized abbreviations that are commonly used, but it's not really important what they are. What matters is that the alleles that any given plant happens to be carrying can be represented by a pair of letters. So a white flowered plant is homozygous for the recessive allele, It's got two copies of the recessive allele that gives it white flower color. And we can write that as two lowercase p's. A purple flowered plant could have two capital P's, two dominant alleles. It would be homozygous dominant. And we could write that as big P, or it could be heterozygous, one dominant and one recessive, and we could abbreviate that as capital P, little p. By the way, if you do problems like this and you work them out by, with pencil and paper, a problem I've seen students fall into is sometimes, especially if your handwriting's a little messy, it's hard to tell the difference between a capital and a lowercase p because the letter forms look basically the same. What I will often do is write my lowercase letter in cursive and block print the dominant. So I'd write a dominant capital P and represent the recessive allele by a cursive P. You do whatever you like. I'm not going to require it anyway, but it is important to keep yourself from making confused mistakes. So this is what Mendel's first cross looked like. The P generation, we seem to use the letter P an awful lot. The first generation that he crossed was between a purple flowered pea plant that came from a long line of purple flowered pea plants. It was purely purple as far as you went back. And the same for the white that he crossed. So he's crossing a homozygous purple plant and a homozygous white flowered plant. And that means that each of the F1s got one dominant purple allele from one parent and one recessive white allele from the other parent, right? The purple flowered parent could only contribute a dominant purple allele to each of its offspring. The white flowered parent could only contribute a recessive allele for white flower color to each of its offspring. So every one of the offspring got a purple allele from one parent and a white allele from the other parent. All of the offspring have the allele combination that we can abbreviate capital P, little p. All of the offspring were heterozygous. Okay, I'm going to introduce a couple of new terms. They're coming up on a slide, but I'll tell you now. When you describe the alleles that an organism happens to have, we call that the organism's genotype. When you're describing the features, the physical traits that an organism has, we call that the phenotype. So the parental generation, you had one parent had the genotype capital P, and the phenotype purple flowers. The other parent had the genotype little p, and the phenotype white flowers. All of the offspring had the heterozygous genotype big P, little p, and the phenotype purple flowers. If I ask you a question on a test, and I ask you, what is the genotype? The answer is always going to be some combination of letters. It won't necessarily be P's. It might be, big C, big A, little a, or something like that. But if I ask for a genotype, it's always going to be a string of letters like this. If I ask you for a phenotype, it's going to be a physical trait like purple flowers or chocolate brown fur or Marfan syndrome, where you get really tall and you have long arms and you suffer from heart defects. Phenotype always refers to a physical feature of an organism. Genotype refers to the alleles that organism is carrying. Sorry, checking the chat again to see if there's anything more to do. So that's what his cross looks like in the second generation, though. We've looked at the F1. Each of those F1 plants could pass on either the big P allele or the little p allele, but it can't pass on both. You can't pass on more than one allele to each offspring, at least not normally. There are cases where it can happen, but that often causes things like birth defects. But if things go as they're normally supposed to go, you only pass on one of each of your alleles, right? You yourselves have a whole bunch of genes. You've got something on the order of 18,000 of them. And for almost all of them, you'll see an exception later, but for almost all of them, you have two of each gene. You got one from mom and one from dad. And if you have kids, you will pass on one of yours to each kid. Normally, you would not pass on both of each pair. You pass on one of every pair. So how can this happen? Well, either if you're crossing two F1 plants, one possibility is that both parents will pass on a capital P, and that will give you an offspring that is homozygous dominant. A second possibility is that the first parent will pass on capital P, and the second parent will pass on lowercase p, and you get a heterozygous offspring, big P, little p genotype. The third possibility is that the first parent will pass on a little p, and the second parent will pass on a capital P, and then you get a heterozygous genotype. And the fourth possibility is that both parents pass on a little p allele, and you get the homozygous recessive genotype, little p, which results in white flowers. Okay, I'm allowing the beauty and simplicity of that to sink in. It turns out to work out the same as if you're flipping two coins, right? If you flip one coin, it can either be heads or tails. So if you flip two coins, you could get both heads, or you could get the first as a head and the second as a tail, or you could get the second is a head and the first is a tail, or you could get two tails, right? There's four possible outcomes for a two-coin flip. It works out the same way here. There are four possible outcomes for one of these F1 crosses. Two of the outcomes happen to be identical, but it doesn't matter here. You can either get homozygous dominant, homozygous recessive, or two different ways of getting a heterozygous plant. All right, we can make this a little bit easier to visualize by using a calculating device that was invented after Mendel's death by a biologist at Oxford University in England, around about 1910, I think, a guy by the name of Reginald C. Punnett. And the only thing you really need to know about Reginald C. Punnett is that he invented something called the Punnett square. Okay, that's all you need to know. Easy enough. And you start by drawing a square. That's a square. Any questions? Okay. Oh, I'm checking the chat. I'm starting to go long. Okay, sorry about that. All right, I tell you what, by the power vested in me by the state of Arkansas, I'll stop here. And on Friday, I'll take you through how to work Punnett squares. I will also, this afternoon, post some practice genetics problems that you can work out using Punnett squares. You don't have to do them. I'm not going to count them as part of your grade. You can take as long as you want to. but they are problems of the kind that are going to show up on the next exams. So it would be a good idea if you were to do them. But they'll give you some hands-on practice in working problems like this. But I'll put those up. But on Friday is when we'll go into how to do Punnett squares. So by the power vested in me by the state of Arkansas, I will stop the recording.